Hi everybody, my name is Dee. I'm a neuroscience and philosophy major, and my Keystone is titled Defining a Barrier in Neuroscience, Spatial Navigation, and my advisor is Dr. Michael Hassimo. So whether we realize it or not, spatial navigation is a part of our day-to-day lives. It's how we get from home to work and work to home, but it's also how we get from our bedroom to the kitchen in the morning to make breakfast. So neuroscience research on spatial navigation really encompasses everything from how our brain represents a physical space, how we're able to move throughout that space, and how our cognitive maps of that space change over time as both us and our memory of that space also change over time. And for my project, I'm looking at defining a specific feature of space, namely barriers. And defining barriers is really a greater part of the mission and understanding of how our brain processes space as well as its distinct features. So my project revolves around the question, what is a barrier? So before I get into anything, what have experiments told us about space so far? The first thing I'd like to bring up is a distinction between what we call allocentric space and egocentric space. So allocentric space is world-centric space, and you can think of that as being similar to a third-person point of view, and it's typically described using cardinal coordinates, so north, east, south, and west, and that's in contrast to egocentric space, which is self-centric, and that's similar to a first-person point of view. So think of to your left, to your right, to your front, or behind you, and I've added some pictures to kind of help illustrate this point. And I bring this up for two main reasons. The first is going to be that this is relevant to a specific kind of cell that I'll be talking about in just a minute. And second reason is going to be because the transformation in your brain between allocentric and egocentric space is really important for how we're able to navigate in a dynamic, constantly changing world. So say, for instance, you're walking down Comm Ave, and you turn because you need to cross the street to get to a building for work or for studying or for whatever it is that you're doing. And you turn, and you see it's blocked off by construction. So your brain is going to take that egocentric visual sensory input that the road is obstructed and you can't cross that way, and feed it in to update your allocentric map of that same exact environment, so that way if you need to take that same route the next day, you're able to successfully remember allocentrically that that route is blocked off, so that way you can replan and reorient yourself to get where you need to go efficiently. So now to neural representations. So this is looking at barriers as represented and defined by the activity of cells called egocentric boundary cells, or EBCs as I will be calling them. And this is data that I helped collect under the supervision of Samantha Malmberg in the Hassimo lab, and this is from EBCs in the retrospinal cortex of mice using calcium imaging. So the way that our experiment works is we will take a mouse, we'll place it in a just empty square open arena, we'll let it run around for about 30 minutes, we'll take it out, and then soon after that we'll reintroduce it to that same environment, except this time with an inserted circumventable wall in that arena. So the mouse cannot jump over it, it can't cross it or climb it or push it around. We'll let the mouse explore with that inserted wall for a bit, take it out, and then return it again to that empty open arena to round out the experiment session. And what we see is actually very interesting. So this is all data recorded from one cell, and you can see those three squares with the colorful dots on them each correspond to the three previous trials that I just described. And each of those colored dots represents a time when this neuron fired. So you can see in the first and the third trials, you have the highest amount of firing activity from this EBC around the outermost walls of the arena. But then when you look at the second trial, when we have that inserted wall placed in the arena, you see a similar amount of high firing activity around that inserted wall. And this kind of difference in data is originally where I got my Keystone project idea to begin with, because it immediately begs the question of why are we seeing this cell respond to what seems like two different features in the environment, and how are they similar? And my plots are a little messed up with the font there, but it's fine. So I'll briefly talk about these kind of heat-looking maps right underneath the square plots. These are called egocentric tuning plots, and these essentially show that the tuning preference of a cell. So that is a spatial relation related to the animal that this cell likes to fire into. So you see the most heat in the map in the color of yellow and green, and that shows that the cell typically likes to fire when a boundary or a barrier is to the left or a little bit left and in front of the animal. And so we can kind of get a tentative definition from a barrier just from this data alone, and I've kind of been alluding to it in the way that I've been speaking, with boundaries being defined as the outermost walls of a space, in contrast to barriers, which you can think of as the inserted wall that we had in that second trial, being defined as an uncrossable obstacle or inserted wall that is separate from the outermost walls of the rest of the environment. So why is this distinction important? Well, the discovery of EBCs in self is fairly new, and there's still a lot to learn about how exactly they encode space and what it is that they're paying attention to. We know that EBCs respond to egocentric boundaries at all kinds of different angles and orientations and distances from animals, but this is what I think is super interesting, is that they're also able to tune to barriers and boundaries that are behind the animal, which suggests that there is more than just sensory input that these cells are responding to. And understanding what those non-sensory cues are really important to helping us see how these spatial frames develop to begin with, and also how they decline naturally with age, and also in pathological cases such as Alzheimer's disease. So what exactly are the mnemonic inputs that EBCs are responding to? I decided to answer this with a functionalist definition. So functionalism is a school in philosophy that argues that what something is based on the role that it plays in a larger system that it is a part of. So for example, we can look at how a functionalist might define a chair. A functionalist might say something like, a chair is what one can sit on without falling off or sliding off. So obviously, the plastic chairs that you're sitting in right now fulfill that definition pretty well. But also under certain circumstances, tables, rocks, boxes, or really anything with a flat, seatable surface could also fulfill that definition of being a chair. And I really like this definition because it's extremely useful in the context of neuroscience and spatial representations, because it accounts for the high level of plasticity and mixed selectivity that we see exhibited by neurons. And mixed selectivity really just refers to the idea that neurons are able to adapt and to play different roles depending on the needs of your brain at the time. So functionalism and context dependency. Cells are known to remap and change their tuning preferences based on their environmental context. And so barriers might be one of those cues for your brain to remap based on the functional context of, quote, what might be just barriering, as we might say, or acting as an uncrossable, immovable object. And this is just a hypothetical experiment that I kind of made up of how we might look to test something like this. So we might run one half of an experiment that is similar to the experiment that I described earlier, but what would we see if we trained the animal to be able to cross the barrier, either by jumping over it or climbing over it? Would we see a difference in EBC activity if the animal was able to cross the barrier? And then again, translating this, thinking about how this would translate into humans, a lot of different interesting aspects that I had thought about in my paper. Maybe the way that our EBCs would respond, or maybe the way that we think about barriers is depending on our individual capabilities if we are working off of that functionalist definition. And then different linguistics, different languages have different ways of describing space. There are some languages that only describe space in allocentric frames. They don't have any egocentric concepts of left, right, or front and back. So how would that affect the way that their neural representations are formed? A lot to think about, as always, in terms of humans, but I would like to thank you all again, as well as my advisor, Dr. Hasimo, Samantha Malmberg, Gianna Matisik, the Hasimo Lab, and KHC for putting this all together. Thank you.
Okay, any questions?
In your, great job, by the way, really great presentation. In your hypothetical experiment where the mouse was able to cross the barrier, do you have like a hypothesis of what you would expect to see from the EBC cells?
Yeah, so if I had to guess at this is based on an activity and papers that have been conducted looking at other cells called border cells, which exist on a spectrum of other kind of boundary types of cells. And border cells work if they specifically respond to things that serve as path obstructions to animals. So there's an experiment run looking at border cells and boundary vector cells where they had a mouse and they had a wall that was suspended about a couple centimeters above the mouse where it could cross under it, and they would progressively lower it to see if they could get these cells to start firing. And they didn't see that border cells and these boundary cells wouldn't actually fire until the path or the wall was fully lowered to obstruct the mouse's path. So I would guess if the mouse was able to cross that boundary in that case, and you'd probably see reduced firing activity or maybe no firing activity at all.
Any other question?
Sorry, I may have missed this in the presentation, but what is it that is giving them the view behind them if it's not sensory input?
Yeah, so there are a couple of different theories about that. I can't really say that I know too well myself. It's still being something that's really studied with EBCs also. But it's thought that perhaps with mice that might be from other sensory input that they're getting, so they're very smell-oriented animals. So maybe it's something about a smell cue or a hearing cue that they're taking in, so another sensory cue, or it's something that we don't really understand yet. Some studies have, sorry?
So what you were mostly focused on was vision.
Yeah.
Okay, got it.
Yeah. Most of these definitions and understandings of cells take input from visual cortex, so that's pretty much the basis for a lot of the data we have now.
Any other questions for Dee? I have one question. Is there anything, did someone raise your hand? You first, girl.
my question was, it's attached to the visual input. Was the barrier that you included in the experiment opaque? And if it was translucent, do you think there would have been a difference in the sensory map?
It's a great question. So the barrier that we did have in that experiment was opaque. It was just essentially like a cardboard wall that was painted white. So in that case, it was very obviously an obstacle. But I would guess that if the mouse was physically foraging around the environment, even if the barrier was clear, you would probably still have some activity from those EBCs because it still is a physical object that it's able to at least get that sensory input from. But I'd be really interested to see what the difference would be in that experiment actually being run. It's a great question.
How much time we have, Adam? What was the biggest setback you faced in this project?
Technical difficulties. I have to say, but that's just that just comes with the lay of the land for lab research, is that the scope we use to do calcium imaging, we have to do a little screw onto the mouse's head to fully attach the recording piece, and that cost a large amount of money that I could never dream of having at this current moment, but the mouse sometimes chews through it or will jump out of the arena, will try to jump out. And so there's just a lot of mouse wrangling that is involved at times, but it's all good.
Anything else? All right, round of applause for Dee.